Hi everyone, am I audible?
Hi everyone, am I audible? Hi everyone, am I audible? Hi everyone, am I audible? Hi everyone, am
Yeah, sorry about that.
Yes, ma'am.
Can you all hear me now?
Yes, ma'am.
Hello, can you hear me?
Yes, ma'am.
Yes, ma'am.
Okay. So then let's get started with the first session of image analysis for biologists. So if you all don't mind, we can start with a quick round of introduction since this is the first class and there may be people from different backgrounds. So I can first go ahead. So I'm Sindhu. I'm a final year PhD student at the Indian Institute of Science. And I work in the bioengineering department and I look at the biophysics of epithelial tissues. And my background is from biology. I have an integrated MSc in biological sciences. Maybe next, Trishna, can you introduce yourself? Just what are you doing now? What is your background?
I audible?
Yes, we can hear you.
Okay, so I am a third year BS student here in ISSS.
Okay.
I have a bio major and I'm currently interning in Siddharth Jhunjhunwala's lab in bioengineering.
Okay, great. Vaishnavi?
Good morning, ma'am. I am Vaishnavi and I am currently pursuing my MTech from in industrial biotechnology, and I have done my BTech from biotechnology from Khwaja Moinuddin Chishti Language University and from like MTech from Delhi Technological University.
Okay, thank you. Next, Divakar. Okay, if Divakar is not there, Manya, are you there?
Am I audible?
Yes, yes, we can hear you.
Good morning. I'm Manya. I'm also a third year undergraduate, and I'm also majoring in biology.
Okay. So let's get started with the first class then. So also you can see my slides, right, on Google.
Yes, ma'am.
Okay. So in this like this course is called Image Analysis for Biologists. So here we will mostly be covering the basics of image analysis, about what things can be done, but we won't really be going into the algorithms of how it is done because this is a basic introductory course. So there are also more advanced image processing courses. Like for example, in this course, we will be learning how to segment cells, but we won't. So there are softwares that have already been built that can do that, but we won't really be going into the nitty gritties of how exactly the segmentation algorithm works. So that is for a more advanced course. This is image analysis for biologists. So this is for people who have biological microscopy data, and they want to do different kinds of analysis with it. If you are interested in more building the algorithms or building softwares that can do these kinds of analysis, then maybe a more advanced course would be more relevant. So in the first course, in the first lecture of this course, we are just going to look at the basics of what is an image, and we will also go into the basics of what is FIJI. So FIJI is like the main software that microscopists use for processing microscopy images. So let's get started. So here, this is the basic outline of the course. So we'll just look at some of the basic things that you can do with FIJI so you get an idea of the software. So what is an image? So an image is, you know, if you just see it, it is just a,
It is not
yes.
visible.
You're not able to see the slides.
No, ma'am.
Now, if it is not in full screen, okay, the screen itself is not visible. I'll share again, sorry. Okay, now it should be visible, I think.
Yes, ma'am.
Okay. Yeah. So if you look at an image, visually it is just a picture, but how do computers see images, right? So computers see images in the form of these pixels. So if you zoom in to an image, like it's the red color dot here, then you see that that small zoomed in portion has different colors. So this is just a simple black and white image. So there are different shades of black, different shades of white, and different shades of gray. And each of these colors have a corresponding number value. So the computer,
Ma'am, again, not like again,
uh,
it's not visible.
it's not visible. Sorry, I think it's maybe I'll try to show it without the slideshow, but somehow the screen sharing is stopping. Yeah, okay. I'll just keep it like this instead of going into slideshow. Yeah. So if you zoom into this particular point, then you see that there are different grids of different shades of black and white and gray. And each of these colors corresponds to a number. So the darker the color, so the darker colors have a lower value and the whiter regions have a higher value. So when you see this is how the computer stores the data of images in the form of these numbers, and each of these number can be considered as a pixel value. So the higher the pixel value, the darker the shade in the final picture you get. So this is the basic of how an image is stored in a computer. So these pixel values can be represented colorfully in different forms. So if you see, this is the original image, and these are all, if you compare all of these four images, they look different, but the underlying pixel values or the storage in the computer is the same. It's just that their representation is different. So if you compare the first image and the second image is a bit brighter, and that just comes because of adjusting the display settings. And similarly, in the third image, whatever white pixels have become black, and the black pixels have become white. So it is essentially an inverted image. And the fourth image here is just using a different color scheme to represent the same values of the pixels. So this in Fiji, how the pixel values are displayed on the screen is you can adjust it with something called LUT or lookup table. So for example, this is a basic lookup table for a grayscale image, which starts from 0 and goes to 255. So this is a very straightforward lookup table where the low values get a darker shade of the pixels. And there are other different lookup tables as well, which we will come to soon. And each image will have a histogram of the intensities. So if you, maybe this I can show with an example on Fiji. So I have already installed Fiji. So in this class, I will just show you some images directly. In the tutorial, you can also work alongside. So I'm just, Fiji already has some sample images, which I will use. Maybe, one minute. Let me just check which sample image I had selected. Yeah, I'll open the sample image fluorescent cells. In the next tutorial sessions also, we will be working with these sample images that are already available in Fiji. So let's take this as an example image here. So this is a multi-channel RGB image. And now if I go to the histogram, so the shortcut is Shift Control C. So you see that for the red channel, there are different intensity values and the peak is somewhere around maybe 50. So this histogram represents the distribution of the pixel values. So it means that most of the pixel intensities are around the value of 50. So now I will just show you the red channel separately. So right now, this maximum slider is at 255. So it means that the brightest shade of red will be displayed when the pixel values are at 255. But now if I move this slider, you can see that the image is getting brighter. So what is happening is when I move the slider, so right now it's at 155. Pixel values that have 155 as the value are also being displayed with the brighter shade of red. And if I keep on increasing it, so right now even the ones that have 75 pixel are being bright. So you can see that the image is getting brighter and brighter. And the other way is if you can also move this slider, so initially the slider is at 0. So I'm moving the minimum slider right now. So whatever pixel values are 0 are having a darker shade. But if I move it, then right now whatever pixel values are at 57 are having a darker shade. So most of the image is dark. And if I go beyond the range of intensity values that are in the image, then eventually the entire image becomes dark. So this image histogram is an important feature of images, and this can be used to adjust the brightness of the image and so on. And this while imaging, so suppose you are also doing some microscopy experiments and you're trying to get an image, then also you can look at the range of values in the histogram. So if the histogram is wide, yeah. So here if the histogram is wide, then it's like there are a range of intensity values in the images and it will be easier for further analysis. So that's about histogram. So I'll be moving between slides and these kind of demos with FIJI in the real time. So this is what I was saying, that this entire histogram shows the range of intensity values, and say at 50, most of the pixel intensity values are 50. So there you get a peak. And when you can move the sliders to adjust the minimum and the maximum intensity values. So if you move it to 80, then even pixels that are at 80 will be displayed bright. So you can see the image better. So this is the example that I told you where I said that all of these 4 images, they look different visually, but the pixel values that are saved with the image are the same. So you can see that the histogram is the same and the other statistical values of the histogram like the mean, median, etc. are also the same. But the same image can be displayed in different formats using these different kinds of lookup tables. So in FIJI, how do you see this histogram is by, I will just show you with the sample image. So you go to image, adjust, brightness contrast, and this window will open. Right now, I already had it open, but if you want to open it, image, adjust, and brightness contrast. And then you can use these sliders to change how the image looks. And here you can also change between different channels. So right now it's in the green channel, and if I move this slider, then it goes to the blue channel. And similarly, you can also change the histogram or the display settings of each channel separately. Another way to do this is you can either change it manually, or here there is this option called auto, where it will automatically choose the optimized display settings when you click auto. Yeah. So this is about histograms. And it also, instead of going like clicking it many times, image, adjust, brightness, contrast, you can also do Shift Control C, that is the shortcut for Windows. And for Mac, the shortcut is Shift Command C. So I think right now some more new people have joined. So before we started the class, we did a quick round of introductions because it's the first class. Those who have joined new, can you introduce yourself? Divakar, are you there? Okay. Diksha? Diksha has left, I think. Neville, can you introduce yourself? Okay, I'll just go ahead with the class then. Yeah, so generally the images that you get from the microscope, if you open it in like normal viewer, like the Windows photo viewer, they would be looking dark like this, because these standard photo viewers, the microscopy images that we get are typically, we acquire data in 16-bit. But if you open them in an 8-bit reader, then the pixel values would be dark. But when you adjust the histogram settings, then it becomes bright and you can see the image properly. And yeah, another important thing to keep in mind about images are how the numbers are saved. So here I told you that the numbers are saved in the form of pixels, and here you can see that the maximum value that it is going up to is 255. So this range of values that the pixel values can take is determined by what is the bit depth of the image. So the simplest kind of image is a 1-bit image where the pixel can only take the value 0 and 1. So you just see either completely dark pixels or completely bright pixels. And on the other hand, you have a 2-bit image. So 2-bit meaning like 2 power 2, so 4. The pixel can take 4 values, 0, 1, 2, 3, and 4. So here also the image has, now it has more shades of gray, but it's not as resolved as the other images here. And in a 4-bit image, then it can take 16 values, 2 power 4. And in an 8-bit image, it can take 2 power 8 number of values. So the higher the bit depth, the more resolution and clarity I would say clarity actually, the more clarity you can get on the image. So these are all again black and white images, but you can also have images that are colored. So in color images, you have, so right now in these black and white images, each pixel has just one value, but in these colored images, each will have 3 different values. So one value for the red channel, um, one value for the green channel, and one value for the blue channel. So, and if you combine all of these 3 channels together, you get a merged image. So typically this is how images from fluorescence microscopy use. Like you may be having different fluorophores. So you might have one DAPI channel, one GFP channel, one red channel, and so on. So you can also work with these multi-channel images in FIJI. So I'll show you some of the basic things that can be done on these multi-channel images using FIJI. And yeah, my screen is still being shared. Okay. So this is the sample image that I have already opened. So right now, if you see, these images have all three, red, green, and blue in them. If you want to see them separately, you can split the channels. And here you can also move between the channels by moving with the slider. So the first channel is red, you can see on the top, and the second channel is green, it's on the top here. And the third is the blue channel. Now, if you want to separate these three images, you can go to image, color, split channels. So now the DAPI image has become separated, the green channel image has become separated, and the red channel image has become separated. So sometimes when you get the data from the microscope, then it's possible that the different C1, C2, C3 channel images are saved as different images. Then you will have to open them separately. And then if you want to then combine them together, you can just go to merge channels. So whatever you need to do for these multi-channel images are there in image and color. And here you can go to merge channels and select which you want as the red channel, the green channel, and the blue channel. And now if I do it, then the image has again combined into one single image. So now, right now, the image is like the red channel is displayed in red color. But say you don't want red color, you want to represent it in a separate color. Then you can, suppose you just want to change it for the representation for the red color. And if you have any doubts, you can just interrupt me in between, because I don't have the team screen on. I have the slides in Fiji. So if you raise your hand, I won't be able to see it. So you can just unmute yourself and speak if you have any doubts. Yeah. So now let's see, if you just want to change the representation of the red channel, you can go to, you'll first have to split the channels. And right now, so say instead of red, you want to show it in a cyan color, then you can go to lookup tables here. So lookup tables is in the main FIJI menu. And if not, you can also go to lookup tables from images. Both of them are the same. Now, if you select cyan, then the representation of the image has changed to a different image. Different color. It's just the pixel values are the same. It's just the representation that we are changing by changing the lookup tables. You can also, so here it is a single channel representation, but you also have other options where it's a multi-channel image. I mean, multicolors. So you can just see what works best for you. But typically these microscopy images are represented as red, green, and blue images because they have a good contrast between them. And even if you merge the channels, you will be able to, so now I changed blue to red. The DAPI channel, the nucleus channel to red, you can again change it to blue. I will again. And before you, so right now I selected this image and then I went to LUT and I'm changing. So this, whatever LUT I'm selecting is happening for the nucleus image. Blue. But if I want to change for this, then just make sure to click and select this once and then go to lookup table and change. But this is still selected. And now, suppose you are using an LUT or a lookup table which has different colors, like maybe this rainbow RGB. So in this LUT scheme, the red color represent the pixel values that are high. So, but then it might be confusing to people who see it. What do the different colors indicate? Like which are the dark pixels and which are the light pixels, because there are different colors in this. So in that case, then you have to use a LUT bar. So that can be added from FIJI itself. And another useful way of representing the image is as a grayscale image. Mm. So yeah, also you can check the bit, how many bits the image has here. If you go to image and type, if it's an 8-bit image, then the pixels will have value from 0 to 255, which you can also see from this histogram here. But yeah, the raw image itself was 8-bit. So even if you change it to 16-bit, the pixel values won't really change. But if you have a raw image that is 16-bit and then you change it to 8-bit, then the image clarity will reduce. So I can then go back here as well.
Excuse me.
Yeah.
So I was wondering, how does the software uh decide that which range of values it would choose a particular color in RGB?
Um, so generally when you open the image, it opens it with like 0 to 255 as the minimum and maximum display value. And if you press auto, then I think it chooses a value for the maximum such that 0.4% of the pixels are above that. So that's how the software decides how which values to be displayed when you click auto. But when you open an image, by default it just displays 0 as the minimum and 255 as the maximum.
Okay, this is for single color, right? I was thinking about RGB, since there are three different colors. How does it decide between the three colors which range of values will go for each color?
So let's say I open this image again as the original RGB image. So right now it's in the red channel. I think even for RGB images, it looks like it's the minimum and maximum are still 0 to 255. And then if you want, you can change the values here.
Okay.
And then it just combines and displays all the three colors together.
All right.
Yeah. So these are some of the basic things that you need to know when you work with um, these kind of multi-channel images. And yeah, one important thing is when you save this. So for example, say you want to separate the channel. So you go to image, color, split channels, and now you want to save these. The format in which you save it is important. For generally, if you don't want to lose any data, then TIFF would be the best way to save it. So you can just go to File, Save As, and then here there are many different formats that you can choose. So you can just save it as a TIFF file. I'll just save it on my desktop now. If you save it as, let's say I'm adjusting the brightness contrast. So if you adjust the brightness contrast and then you save as TIFF, please. And now let me try to open it again. So when you save it as TIFF after changing brightness contrast, the original data is still there. Like you can still change the intensity values. But say now you save it as JPG, then it just saves the displayed values. So I've saved it as JPG. And now if I open the JPG image, so the other older values we had has changed now when you change it as, whereas if you compare it with TIFF, ideally this is how it should look like, but in JPG it just saves whatever was displayed and not all the pixel values. So it compresses the image and that leads to data loss. So whenever you save images, always make sure that you save the raw image, which is mostly in this TIFF format. When you change it to JPG, it changes the underlying pixel values itself. So JPG is not a very good format for saving images. But say you want to put some of these images in presentations and so on, then JPG would be a better option because you can select what brightness settings you want and it saves it with the brightness settings that you have selected. But always make sure that you have the raw TIFF file as well with the correct pixel values. So this is about how to use multi-channel images. There are also again different settings that you can play around with if you go here. But right now I've just shown you the most important things that you will be needing often. And in the tutorial sessions also, we will be using the same sample images that are already there in FIJI. So this was about multi-channel images. You could also have 3D images where each plane is like, for example, if you have used a microscope, you would understand it better, that if you have a 3D sample, then you can click one image per Z position in the sample. So you'll have a series of images or a stack of images. So for this, I'll just open a sample image first. Confocal. So here you can see that there is a Z slider which you can slide through. So this is the first Z stack or the bottommost image. And then as you go up, you're just going up in Z in the sample, like the height of the Z plane is increasing here. So so this sample image has opened directly as a stack, but sometimes each of the Z plane could also be in different stacks. So then in that case, you will have to convert the images to stacks. So I'll maybe show you with an example. So right now it is a complete stack and I will save it as separate images. So you can go to File, Save As, Image Sequence. I'll just save it on the desktop. So now when I save it as an image sequence, each Z will be saved as a separate image. So it should be here. Yeah. So right now, because when I opened the sample image, it opened everything together as a stack, but when you open these microscopy images, then it has also saved them as separate channels. So for example, here you have C1 Z0 and C2 Z0. Now first we will have to put these two together using merge channels. The red would be C1, so you can select C1 here, and green would be C2. I hope my screen is still shared. Okay. So now this is the first Z0. And now you also have Z1, C1 and Z1, C2. Now again, we can merge these two channels. And red is C1, green is C2. Now you have two different RGB images. And if you want to make them into a stack, you can go to Image, Stacks, Images to Stack. I think both of these are so right now these images are already in a stack. The stack is here for the different channels. That's why it's not again making these C stacks. Or you can so if you convert it to RGB, then instead of being represented as two different channels, you get them together. Now the Z1 and Z2 have combined into a Z stack. So this is one basic thing like that you would be doing often, is converting images to stack and stack to images. I'll open the sample image again. So, yes. Yeah. So right now it's in a stack. You can also do image stacks, stack to images, and it will, each Z stack will be opened as a separate image. Yeah, and one more thing that you can do is, I'll just close Fiji because too many things are open now. So say you have a 3D image, but how will you represent it in one single image? So one way to do that is by using Z projection. I'll show you. So Z projection means that it will represent a 3D image as a 2D image. Let me open the same sample image. Confocal. So now there is like some amount of data in all of these Z stacks, but you want to represent it all together as one image instead of having so many images here. So for that, you can go to image. So for these Z stacks, whatever you want to do is in this image and stacks menu. And previously we saw that for multi-channel images, whatever spacing settings that are needed are in the image and the color menu. So here you can go to Z project, and there are different methods of doing the Z projection. So you have average intensity. In this, what it will do is it will compare all the Z stack images. And so for example, if at this pixel, at this point, at Z1 the value is 0, but at Z2 the value is 2, at Z3 the value is 4, then in the final image it will just take the average of 0 to 4 and it will display. So like that it will do for all the pixels in the image. It will just average out the values across all the Z stacks. So you can do an average intensity projection if you don't, like if you want to represent the data, how the intensity distribution on an average. Now another way is to do it with the max intensity. So in max intensity, for each pixel, the FIJI will see which of the Z stacks have the maximum intensity, and that will be projected. So max intensity you can use, suppose you have an image which is has one best focal plane at which the intensity is maximum, but in the other planes, other Z planes, the image is out of focus and the intensity is not maximum. Then if you just want to get the Z plane which has the best focus, then you can use the max intensity. And similarly, another way is by using min intensity. So just like max, instead of considering the maximum value, it will consider the Z stack in which the intensity value is the minimum. And then you can, there is another way where it will just add the pixel values from all of the intensities. So we can also try these different things. This has 25 Z stacks, and there are 2 images per Z stack. So there are a total of 50 images. But yeah, the number of Z stacks are 25. So from 1 to 25, I'm doing an average intensity projection. And this is how it looks like. But if I do a max intensity projection, it will look, the image will look like more in focus. Image stacks. Now I'm selecting max intensity instead of average. So in this one, it has just selected whichever has the maximum value. So the image is overall brighter. And if you select min intensity, then you can expect that most of it would just be dark. And in min intensity, it considers the minimum intensity value of both the red and green channel. So the final image is not a color image, it's just a grayscale image. So yeah, so generally the takeaway here is that if you want to select, like if you have an image with one best focal plane, then you can go for the max intensity. But if you have an image where like the different Z stacks have, say the sample itself has different information in the different Z stacks. One common example is when, example, you're imaging F-actin, then the bottom Z stack will have information about the stress fibers that the F-actin forms, and the top Z stacks will have information about the cortical actin, because actin can exist in different forms and they are localized in different regions of the cell. The stress fibers are in the bottom of the cell, and if you go above, if you just go above in the imaging plane, then the cortical actin comes. In that case, if you do a maximum intensity projection, then you will, and if the stress fibers have a higher intensity, then you will lose information about the cortical actin. But if you do an average projection to represent that kind of data, then in the average projection, you will have both information about the stress fibers and the cortical actin. So you won't lose any information. So which Z projection type that you use will depend on which data type that you have. So far we have been seeing these Z-stack images as a series of images, but you can also see them like right now, you can just see the XY plane, and to see different Z planes, you have to move in the slider. But you can also see the Z plane as a height using the orthogonal projection. So it's just again viewing the same data, but in a different way. So image stacks, and here you can go to orthogonal views. And here you can select which part exactly you want to select. So for example, if I choose this region here, yeah. So for this particular XY point where these two are intersecting, you can see the image here. This axis is the Z axis. So if I move in Z, you can see that this line is moving. So this bottom part is Z is equal to 0. And as you go up, you can see that the line is also moving. So this kind of a visualization is important when you want to see in which height of the cell is a certain protein located. For example, you can see that the green protein in some parts, it is located in the basal region or the bottom part of the cell. And in some regions, it is located at the top part of the cells. So this visualization is like helpful when you want to see in 3D where the protein is located, because in just this 2D image, you will just see in an XY region where it is the protein or whatever you're imaging is located. And actually, there are also options where you want, if you want to view it as a 3D volume. This image may not be the best to show it, but I can just show it. Again, there is one more important feature in FIJI is called plugins, which we will cover soon in the course. I think this does, let's wait. So this, what I showed you before, was an orthogonal view. You can also have a volume view where you see it as an XYZ volume section, but this plugin is a bit slow. If you want to see for 3D images, there are also other good softwares that you can work with. Like one of it is called Imaris. So Imaris has two versions, Imaris and Imaris Viewer. Imaris Viewer is the free version, and there is a paid Imaris version as well. So this 3D viewing, I'm just showing you with some data that is already there. So this is a HeLa cell in 3D. Like you can see that the nucleus is somewhat in the middle of the cell and so on. And with this also, you can again have the orthogonal section view, where if you select a point in space, it shows how the value changes in Z. So this software you can use when you have like very high dimensional 3D data. There generally this does a little better in rendering 3D images compared to FIJI. This is still a cell that is in 3D, but if you have, I can show you with some data that I have. So again, here also you can again see the histogram, and you can modify it. So this is similar to what we saw in FIJI here. So in this sample, you can nicely see that here the Z is high, that the cells are in a different plane compared to over here. And similarly here, the cells are in a different plane. And if you see it as a 3D view, you can see that. So this is the bottom part. I have rotated the image. So these cells are a little bit like in a different plane compared to these cells. So this is the volume view of the image. If you have any tissue sections that can form different 3D shapes, then this kind of a 3D viewer software is helpful. And again, this is just to see how the Z values are changing at a specific point. Viewer, it can only be used for visualizing the images, but the complete Imaris software is expensive and you can also do a lot of analysis with that. Right now, I just wanted to show you this because we are talking about 3D images. FIJI also has these kind of 3D viewer plugins, but they are quite slow and right now it's not responding. I'll just restart FIJI. Okay. There are other options as well to show 3D images, but generally the rendering is better in Imaris viewer software. So you can go for that if you want good 3D visualizations of your sample. Let me open the sample image. This was the image. Meanwhile, do you have any questions or should I just keep going? If you have any questions, you can just unmute and talk directly because I'm not on the Teams window. So if you raise your hand, I won't be able to see it. Yeah. And now, suppose you have, like right now, this data has 25 Z-stats. But it's possible that you have some data that is very heavy, like maybe you have 100 Z-stacks of data because the sample itself has a height of 15-20 microns. So then you may need more Z-stacks to see it. And if there are too many, if the data is very large, then it slows down the software and so on. So then in that case, if you want to coarse grain the data a little bit, or suppose you just have, you don't want the data on the top regions, you just want the bottom few slices, then you can make a substack using this image stacks and then tools here.
If you're sharing your screen, it is not visible.
Okay, sorry. I think whenever the internet is slow, it stops screen sharing. I'll just share it again. Okay, I think now screen sharing is back on. Yeah, so now you only want the first half Z-stacks and not the second half. Then you can just make a substack. Make substack. Or if you also want to remove one channel, because this has both red and green channel, you can use this. So say you just want the red channel and the first 12 slices, then it will just give you that. And there are also other options that you can do. Tags, tools. Okay. Interleaver is when you have two different images and you want one image after the image. Now, if you want, like if you don't want all the Z stacks, 1, 2, 3, 4, but if you just you want to leave one stack in between, maybe you have imaged at a very high resolution. Now, for the analysis, you don't need such a high resolution data. Then you can remove the slices in between. I think I will just, let me close this one. Let's just have the red channel Z stacks. Again, here just initially it was showing it in a gray LUT, but now it is showing in a red LUT. You can use either the styles remover or keeper. So now initially it had 25 Z stacks, now it only has 13. It has removed every second Z stack. And now suppose you want to see all the Z stacks side by side, then you can view it with this montage option. So again, whatever you need will be here in stacks. Make montage. So because there are 25 slices, it will make a 5x5 image. So here it starts from Z0, Z1, Z2, and so on. This is if you just want to see how much difference there is between the Z stacks in one go, then you can use this option. So for these kind of 3D images, yeah, I think I got disconnected. Yeah, and then you can have 3D images with both Z stacks and different channels, like this example we were seeing. And another type of imaging data that we typically get are time-lapse images, where the same sample is imaged at different time points. So for this, we can use this demo image. Yeah. So this is the image at time point 1. And over time, you can see how like these particles are moving and dividing and so on.
The screen is not visible.
Oh, okay, sorry. Yeah, now it should be visible. I think there is some internet issue here. So whenever the net is slow, it just stops sharing automatically. So anyways, I had opened this sample image. Samples, this one. And this is a time-lapse image, meaning that it is the same position that has been imaged at different time points, and you can see how this particle is moving and splitting and so on. So again, time-lapse images, when you open in FIJI or the microscopy data that you get, it will not open as a stack. The microscope will acquire each one image per time point, so it will be saved as separate images. So I'll just save it as separate images. So here, this is just, this will just, you're just telling the software what file name to use when you're saving it. I'm leaving it blank and I'm just selecting 2 digits here. So it will just be saved as 00.if, 01.if, 02.if, like that. And if you want to save it in a different format, you can select that as well. Right now I'm saving it as diff because I don't want any data loss. And here you can select which folder you want it to save. So it should be saved here now. Now, if you want to open many, oh, sorry, it got, yeah, I think now my screen is visible. So typically when you get data from the microscope, you will have images like this, like one image per time point. And if you want to open all of them together in FIJI, you can just select all of them and drag them into this FIJI window here. So then it opens each image, all the images one by one. And once all of the images open, then you can go to stack and make it as a stack. While this is opening, okay, it has opened. So either you can go to, so stacks, you can either go from image and stacks. This will have all the options, or stacks is also present here. Like some of just the important options of stack are here. So you can just do images to stack, and now it has become a stack. And right now, if you want to set how much time period, what is the imaging interval, or between 2 images, what is the time difference, then you can go to Command Shift P or Control Shift P. Here you can set the parameters for the image. So this just has one channel image, so 1. And these are not Z stacks. So it just has Z will be 1, and but the stack is in terms of the time frame. So there are 50 time frames. And now let's say this was imaged in a 20x objective. So this value will change based on the microscope and the objective that you use. But typically for a 20x, it's around 0.325 micron per pixel. So here you can set all of the scaling factors as well. So maybe this I can include in the slides. So when you set the image properties, you will have to know what is the pixel to micron conversion. And this will depend on the specific objective and the specific microscope that you have used. But generally for 10x, it is around 0.16 micron per pixel. And for 20x, it's around 0.32 micron per pixel. It might be a little different for different objectives and so on. And for 63x, it is around, before that we have 40. So anyways, I'm just using these values roughly here. May or may not be correct because these are some sample images about which we don't know much. And this voxel depth is the Z imaging interval. So suppose you take Z stacks every 1 micron, then this value has to be 1. But here it does not apply because we don't have any Z slices. And let's say that these were imaged every 1 minute. So this ideally you will have to do it for all the images that you get. So even in the, sorry. I'm sorry about the screen sharing. I'll try to figure it out. It's just stopping sharing on its own. So yeah, here I was just showing you how to set these values here. And this has to be done even for this image. If you see the scale bar or the conversion from micron to pixel is already kept, like it's already in micron. For the image is a 400 bar 400 pixels, and in microns it is 21.78 cross 21.78. So in this image they had already set the micron to pixel conversion factor. And again, the Z interval is also about 0.05 microns. So this, when you do more analysis on these images, it's important to set these micron, pixel to micron, and the time interval values correctly. And for these kind of time lapse or stacked images, whatever brightness contrast you set for one image, that will automatically be set for all the other images in the stack as well. So this is about time-lapse images. And then we have something called as ROI manager. So let's go to this time-lapse image. Okay, sorry, I'll just open it again. Or we can also just, I can just show you with the confocal image. That is also closed. So you can use this if you want to measure the intensity values or the shape of a cell, etc. I think this would make sense if there are any. Let me see if there are any sample images where the cells are visible nicely. Yeah. So, for example, here you want to look at what is the red intensity per cell, what is the green intensity per cell, what is the size of the cell, what is the size of the nucleus, things like that. Then you can use, like, select each cell as an ROI. ROI means region of interest. So you can, if you select this option here, then you can just draw a boundary around the cell. Right now, I'm just drawing it roughly. and you can save this as an ROI. So the shortcut is, once you finish drawing this, if you select T, it gets added to this ROI manager here. So this is the first ROI. If you select it, it will come up here. And if you want to show it with the labels, you can also do that. And now this is the second cell. Again, I'm choosing this drawing tool. Here it looks like the whole cell is not in this frame of view. Some of it is outside as well. But I've just selected whatever is within the image and I've added it with T. So this also is added. The first one is added. Now the third one. Sorry. This would be easier if you have a mouse. And similarly, the fourth one. So right now, this I am drawing manually by hand to get the cell outlines. But so this is a kind of segmentation, but it's manual because I'm just doing it by hand. But later in the course, we will also see how we can automate it, and there are softwares that can automatically detect these cell boundaries. So these are the 4 cells, and if you want, we can give them better names. Cell 3 and in this, for each ROI, the software can calculate whatever you set here. So if you go to measure, then for each of these, this is the first ROI or the first cell, second cell, it has calculated the area. And by mean, here is the mean intensity. So right now it is a merged channel image. So it has calculated the mean intensity for red, green, blue, all 3 channels together. And then X and Y are the centroid coordinates. And then you also have the perimeter. And this major and minor axis are, it fits the outline that we draw to an ellipse and gives the length of the major and minor axis and so on. But if you want to, so right now we have these ROIs. And if you want to save it for future analysis, then you can save it as well. Actually, you have to select all the ROIs, then it will save all 4 ROIs. Yeah, then it saves it as a zip file. ROI. No. Yeah, so now it is saved. So next time, if you want to analyze for something else, you don't have to draw it again. Now, let's say we just want the green channel intensity, then you can split the channels. And now on this image, you can. So these ROIs can be applied to whatever image that you want. So, sorry. So now again, if you do measure for these 2, for these 4 ROIs, then again, you get So actually we have 8 values because we had the first 4 values. It's all getting appended to the same frame. So you can close it. And if you want just these 4 measurements, now you just have these 4 and you can save it as a CSV file later if you want to analyze it. Yeah, right now I'm just closing it.
Ma'am, the screen is not visible.
Oh, sorry. Yeah, so sorry about that. It's just randomly stopping the sharing. Yeah, so now we have split the channels, and now if I do measure, it just gives you the mean for the green channel. So these kind of analysis, if you want to check if um, cells that are smaller or cells that are smaller have higher green intensity levels, then you can do these kind of analysis. But right now in this image, it does not have many cells. It just has like 4 cells. So you can manually draw the boundaries and do these analysis. But there can also be images where there are many cells, like hundreds or even thousands of cells. There it would be difficult to draw the boundaries one by one, and that's when we go for segmentation. And yeah, so far. And now these ROIs that I saved, yeah, you can also open it in Fiji. Like you just have to drag and drop, and then these values are saved here. And you can also label them and so on. So it's been almost 1.5 hours now. So we can take a quick 10-minute break and we'll be back by 11:30 and we can continue. Okay. Because it's a 3-hour lecture, maybe like in the middle of the lecture, we can take a 30-minute break and we can reconvene at 11:30. Okay, I hope all of you are back after the break. Yeah, so in the chat box, I had shared a Google Drive where the lecture slides will be uploaded. Yeah, but I would say that more than the lecture slides, it's the during the lecture itself, I will be showing how to do different things in Fiji. So the lecture itself would be more useful. But if you want to recap what was taught in the lectures, then you can have a look at the lecture slides. So till now we were at how to use the ROI manager and the measure function to analyze different cells or other structures within the images. So now let's look at some of the other basic things of using FIJI. So again, I'll use some example image, maybe this one. So now you can, so earlier we were drawing the ROIs with this freehand selection, but it can also be drawn with these tools as well. Again, similarly, you can draw with circles or and so on. And if you want to measure the length of an object, like maybe not exactly, but for example, you have a microscopy image. This is actually not. So first this is still in pixel. And if you want to get a rough estimate of how to, what is the diameter of the nucleus, then because it's in pixel, I will first set the scale. It's a single channel image. And let's say this is, I don't know, again, it looks like a 40 or a 63x, but because we don't know the exact values of the sample image, I'm just giving a rough estimate.
Excuse me, you're not sharing the screen.
Okay. I think the issue is with the Wi-Fi. I'm using the ISC Wi-Fi. That's because I tried different things. Yeah, so I just opened this image. And now let's see if we want to get a rough estimate of what is the diameter of the nucleus, what to do. So the first thing that we have to do is to set the pixel to micron scale here, because right now it's only in pixel. So I've changed it with the scaling factor. So now you can see that it has also come in microns. And now you can just draw a line. So if you press, right now I'm zooming in the image by pressing plus on the keyboard, or you can also use this magnifying tool. And now I just draw a line from one end just by if you select this tool and just drag it, this line will draw. And then you can measure it. So for measure, the keyboard shortcut is M. So if you press M, you get the length of this line that you draw. Now, similarly, you can draw for the other nuclei as well. And right now, I'm just moving across the image by using the trackpad. But if you want, you can also use this tool, and then you can just press on the image and move across the image again. So by this you get the diameter of the nuclei. And now again I have zoomed out, and if you want to do any analysis with this, then you can save this as a CSV file. Yeah, but right now I'm not going to save it. And apart from this, you can also crop an image. So just draw a box. So you can go to image and then crop it, or you can also use the shortcut, which is, so now this is a cropped image. And now I just did undo, Control Z or Command Z. That is for undo. So I can show you again. So right now I'm going to crop it with the keyboard shortcut Command Shift X or Control Shift X. But now say I want to go back to the original image, then you can just do undo with Control Z or Command Z. It's stopped sharing again. Yeah, and then if you want to just I think this, I can show you this with the green image. One sample. Hila. Yeah, so suppose now in this image, I want to count how many of these green structures are there. These are sample images. If you want to know about them, you can just, I is the keyboard shortcut, or you can also go to image and show info. So here they have given a description of what is the image, what the red channel is stained for the lysosomes and the green channel is stained for the mitochondria. So now suppose in this image we want to count how many mitochondria are there per cell. Then it would be difficult to, one way is that you just use this tool and draw each mitochondria. That would be very hectic because there are many in this image. So then you can use this point tool, and for whatever you select, you can place a point there. Or if you want, so again, now I use the keyboard shortcut Shift Command Z. You can also go to image, color, channels tool, and you will get this box. So here you can decide which channels should be on or which channels should be displayed. I think the red channel will be easier to work with. So let's say we want to count the number of lysosomes per cell. Now I have selected this point tool, and you just keep a dot wherever you see a lysosome. And finally, you will be able to get a count of how many lysosomes there are. If you count manually, then you might lose track of which ones you have counted, which ones you have not, and so on. So this point tool can be used for that. Something like this. Again, these are all manual methods of analyzing images, which, as you can see, it's quite time consuming. But if the image quality is good enough, like right now in this image, if I just move across, there are some red spots which are also out of focus. But if the resolution was good enough, then we can also do an automated segmentation instead of, doing counting things manually. So yeah. And there are also other tools that you can use. And then this is the text tool if you want to write any text on the image. for example, sorry, red channel image, and you want to label it. Have I selected this? My slides, okay, my screen is still visible. Yeah, also I think I didn't show you how to keep the LUT bar along with the image. So now this, I'll just show you with the red channel of this image. And now say this red channel, I'm changing it to a lookup table which has multiple colors. Maybe this one. Now if you want to put this in some presentation or some report, then you should also, along with the image, you should have a mention of what each of the colors represent. So that you can go by going to tools, analyze tools, and calibration bar. So this will give you the entire lookup table. And I think it might be, right now this whole image has cells. So lower right, there is some gap. Maybe you can keep it here. So now when somebody sees this image, they will also understand what is represented by each of these colors. Yeah, so when I was talking about multi-channel images, I forgot to tell about this. And now say I have this green channel image. So here, if I go to info in this sample image they have used, I will share again. So in this green fluorescent cell sample image, they have not really described what the colors mean in this info file. So we don't know what exactly the green channel is, but say this is the blue channel and it looks like the DAPI or the nucleus channel. And I want to label it like right on the image, then you can use this text tool and just select where I want the text. And now I just write nucleus. Right now the text is in black, so you can't see anything here. But if you double click this, then here you get more options about the font color. So I'll first change the color from, I'm changing the color to white so that it's visible. And then if you want, you can make it bigger. And you can also press it and move it to wherever you want. So this tool is used for that. And then this is just the magnifying tool. Like if you just select this tool and click, it magnifies. And right now, yeah, like you can just press, instead of this magnifying tool, the keyboard shortcut is also minus and plus. Will also zoom in and zoom out from the image. And this is to move around the image. This will be easier if you have a mouse and not a trackpad. Like if you have this selected, then you can just click on the mouse and move through the image. Right now I'm on my laptop on a trackpad, so I'm not able to do it. What else do we have? Yeah, but for day-to-day use, these are the tools that you might be needing. And then again, you have stacks. It's a shortcut. You can go from here, or you can also go from image and stacks. And then you can also select lookup tables from here and so on. And one more thing that you should know is some of the microscopy files, they are not in the form of .tif. Like for example, if you have, I can also make a, so depending on which microscope you use, the format of the file itself would be different. So for typically these Zeiss microscopes, Zeiss is the name of the brand. My screen is good. There, the format is .czdi file, and these can be opened with the Zen software, but it can also be opened with FIJI. So this Zen software is the software that comes along with the Zeiss microscope itself. So it's not that all the data that you have may not be in the format of TIFF file. CZI if you have a Zeiss microscope, and if you have a Olympus microscope, it will be in .oir. Again, this will need the Olympus software to open, but it can be opened in FIJI as well. What else? And then another popular microscope brand is Leica. So in Leica, the format is .lif. And to open this also, you will need either the software that comes along with the microscope, or you can open it in FIJI. So typically, if you have, for example, a Zeiss microscope, then it's better to save the raw data as like .czdi, because this format will have the images and as well as the metadata. So the metadata will have things like which objective was used for the imaging, and which LEDs or which lasers, depending on whether it is epifluorescence microscope or confocal microscope. It will also have information about which LEDs and which lasers were used, and what was the, again, along with this, you will also have the information about excitation and emission wavelength. If it is a multi-channel image, then there will be different sets of excitation and emissions. And it also saves things like, so when you have a microscope stage, then you will be imaging different parts of the sample. So the coordinates of the imaging will also be saved, and so on. So all this metadata, you might be needing it later on in your analysis. So it's better to save the raw data as the .czdi file. And once you have this raw data, if you want to do any specific analysis, then you can export it as .tif for all the analysis that you want to do. If you want to, for example, quantify fluorescence levels and so on. But you can export it as .tif. And if you want to put it in any presentations with the histogram display value set, then you can export it as .jpg, and this can be used to for representing the image. But this data, make sure you never delete this raw file. So suppose you may think that from this will be, the data will be higher, like it will take more storage space because it has all the metadata as well. So you might think that from the CZI file, you open it in FIJI, and then this JPG format may have lesser storage space. So you might think that you can export to JPG and delete the raw data, but please don't do that. Then all the important pixel information from the image would be lost. I'm sharing my screen again. So always make sure that you have at least one copy of the raw data. It could be either this CZI, and even the TIFF files will also have some of the metadata stored. Like TIFF may not have information like the excitation and emission wavelength. These information will still be present only in the microscope raw data file. The TIFF file will have some metadata like what is the pixel to micron conversion that would be present. And also the image pixel values will be preserved. It won't change. So if you want to do any quantitative analysis, TIFF files are good. But in .jpg, the pixel values itself would change. So always keep a copy of this, the raw files. Even if you are exporting it to JPG for something else, also keep a copy of the raw files. So this is very important. And so generally these other kind of files, like TIFF file, you can just drag it into the screen and import, like I already showed. So say you want to open this time-lapse image. You just have to drag it here. But when you open these CZI files also, it's the same. You have to drag it into this FIJI window here. But after you drag it, it will ask for more information about how the file has to be opened, whether it has to open all the channels, all the Z stacks, all the positions, and so on. So I can show you how to open with the CZI because that is the raw data that I have. For now, I can show you with some data that I have. But also, yeah, again, it will be different. If you have a different microscope, like if you have Leica, then it will be a .lif, or if you have Olympus, it will be .oir. I think I have some OIR and some CZI files that I can show you with. Generally, these, what I do is the CZI file raw data copy I keep in the hard drive. I mean, it will depend. Like you can also do it as per your convenience, but whatever the raw data files, I will just keep it in the hard drive or in some place and not disturb it. And these TIFF files exported, that I keep it on my laptop or wherever we will be performing the analysis. So if I don't know if I have many CZI files on my laptop or not, because mostly I export it to TIFF and then keep it in my laptop, and these TIFF files I use for the analysis. Yeah, see, all of it are whatever raw data I have already exported it to TIFF. So I don't have much. Maybe in the next class I will have copy some raw data. Okay, I have one CZI file, but I'm not sure what experiment. Okay, let's see. So yeah, so when you import a CZI file, it will ask for options like this. So if you do split channels, then the RGB images will open as separate red, green, and blue separately. But if you don't do it, then it will open as a merged image. And similarly, if you do split time points, then each time point will be opened as a separate image. But if you want it all to open as one stack, then you can just leave it unticked. And then here series, I will show you. So in these raw microscope files, like there are different positions, because when you do an imaging experiment, you image the same sample at different, there may be 2-3 points. So each of this here is one different position from the same sample. Now if I just want to open the first two, then I'm just opening these two. And here it is also showing information about that there are two different channels in this image and two time points. So if I open, then it will open these first two images. So these are like, this is the first channel, which has the cells, and this is the second channel which has fluorescent beads. And you can see that because I opened it from the CZI raw file, it has already that metadata about the pixel to micron conversion is already there, and it is a high resolution 16-bit image. And it is also has 2 time points. You can see that between the first 2 images, the cells have moved. And similarly, these are some fluorescent beads that were also imaged for this experiment, and that has also moved a little bit. So yeah, it's quite straightforward to open it. Like, you just have to, this is a time-lapse image. I think this is a multi-channel image. Is it okay? Again, I wanted to open all the channels together and all the time points together. I'm sorry, the internet is bad, so it just stops sharing on its own. So previously I had opened one CZI file which had time-lapse data. This is something which has RGB images. So you can see that this is the first green channel. This is the second blue channel with DAPI, and the third channel with red. So opening the CZI or LIF file is the same. You just have to import it, and when it asks for options here, you will just have to select how you want the image to open. And it also has different Z stacks. So here, if you see this, these are just some example images that I have, like some of my data. I'm just showing it now. So here, this is red channel is stained for F actin, and you can see these lines. So these lines are the actin stress fibers. This is the bottom of the cell. And now if I go up in Z, you can see that you don't really see these lines. Now the actin is more near the cell junction. So this is the cortical actin. So this is the example that I was giving when we were talking about making Z projections from 3D image. So in this case, if I do an average Z projection, that would be better, because all the Z stacks are having a different kind of information. But if we do min or max intensity projection, then we will lose information about some of the stats. And yeah, let me check if I have any. I might have OIR also. Some of it. So OIR is the format for Olympus microscopes. Okay, I think maybe it's in my hard drive somewhere. I stopped sharing again. Sorry. LIF. Okay, LIF, I have some old data from 2022, but it's on OneDrive. Okay, I'll try opening it. If it does not open, then in the next class, I can download it and keep it on my laptop. This will take a while to open because it's on cloud. Let's see. Meanwhile, does anybody have any questions until this opens? Also, before we started the class, we did a round of introductions because this is the first class. So maybe those who have not introduced yourself, can you introduce? I think Deeksha, are you there? Who else? Divakar? Are you here? Okay. Anirudh, if you're here, can you introduce yourself? Okay. Okay. I saw your message on chat. Yeah, I think this .lif, it's on. Okay, it has opened. It was on OneDrive, so it took a while to open, I think. And I think this is some image from an immunostaining experiment. So you have 4 channels and 16 Z stacks. So you can select, and there are different positions that were imaged in the experiment. And this is an 8-bit image. And you can see that initially the image appears dark because the visualization settings is at 0 to 250, but most of the pixel values have a much lower value. So I'm moving the slider here. And now if I go up in Z, you can see again similar kind of actin stress fibers. And now this is the DAPI channel, the nucleus channel. And right now all of it has, the lookup table is in red. That's why all the channels are in red. But you can also change this visualization by making it blue. And now all of it is blue. So this is one of the, 1 minute. I'll just be back. Give me a minute. Yeah. So then it's changing for all the channels. So one way to do it would be you can just split the channels, change the LUTs, and then merge them again. I'll just close the other stuff. And yeah, one more thing you can do is, instead of every time you have to, so FIJI is a very click based software. Like you just have to move your mouse a lot. One way to deal with this is you can set shortcut keys for each of the function. Maybe that we can do in the next class, where we also talk about plugins and macros and how to make things easier or faster to work with FIJI. So this is just the phase contrast image. So for phase contrast or bright field, this is bright field actually, not phase contrast. Typically we use a gray LUT, and then nucleus is blue, so that can stay. Maybe this can be the green channel. This, yeah, these are all immunostaining images. And this is the actin channel for which I will give red LUT. So again, I'm just showing you what this histogram can do. So this histogram is a value which, histogram of the pixel intensity. So here, most of the pixel values are at around 100 to 110, I would say. So that's why there is a peak at this point. And if you move the slider, it determines what pixel values are displayed. So right now it's at, the maximum is at 255, meaning that only the pixel values which have 255 are shown bright in the image. But if I move this, right now it's at 128. So even the pixels which have a value of 128 get a bright color. So if I reduce this, it becomes brighter and brighter. And similarly, this is the minimum slider. So right now it means that whatever is 0 has a value of black, or is displayed as black color in the image. I'll share it. But if you move it front, Then, so right now I've moved the minimum slider to 100. It means that even if the pixel has value of 100, it will still be shown as black. That's why it looks like most of the image is black, but the pixel values are still non-zero. It's just that the display settings that you're changing here. So this is about how to open images from other softwares. Yeah, so this was about the first class. It was mostly just some of the basic things that we use in FIJI. Oh, okay, you're also working in IAC, Anirudh. Okay, nice. Yeah, so since we have some more time, I can just quickly go over what we covered today. Also, yeah, and maybe at the end we can then discuss what all will be covered in the course. So we started the class today by generally talking about the basics of what a pixel is and how do computers see images, because when we see images we see it with our eyes as a picture, but computers see images as a group of numbers, and these numbers are what we call as pixels. And these numbers can be represented in different ways. So here these numbers are represented as a grayscale image, where lesser the brighter pixels are represented by high values in the numbers and so on. But even because the pixel values could be the same, but the representation of the image can be different depending on how you choose to display the image. So all of these four images have the same pixel values. So the computer will have the same values stored for these images, but it's just that they are displayed with different LUTs. So LUTs are lookup tables which determine how the pixel values are mapped to which colors. And then we saw, I'm just going a bit fast now because we already covered it. It's just a quick recap of everything we did in the class today. And then these pixel values can be shown as a histogram. And using this histogram also, you can change the display settings, where you can choose the minimum intensity that will be displayed on the screen and the maximum intensity that will be displayed on the screen. And then these are again the same four images with, you can see that their histogram and the statistics of the pixels are the same, but their visualization is very different. So if you compare the first and the second image, in the second image, the image is brighter because you can see that the values are about 1000 is getting the maximum brightness, but in the first image 1000 is only getting a gray color pixel. And then the third image is an inverted image, meaning that whatever pixels are white in the first two images are shown as black in the second image. And then in the fourth image, the lookup table is completely a different color bar that has been used. Yeah, and then we saw how to do this in FIJI. So the shortcut is Control-Shift-C or Command-Shift-C. And then you can see this histogram. If you forget the shortcut, then you can also go to Image, Adjust, Brightness, Contrast. Again, you will get this and you can change the display values here. So here it has changed the display for the entire stack. But in the later stacks of the image, it would be like, this is an oversaturated image because all the pixels are, like, there are many pixels that are greater than 14 and they're all shown as red. So you don't really see the information in the image. So I'm moving back the slider now, and now you can see the image better. My slides, yeah, okay. Yeah, and then these numbers can have a range of values. So, and that depends on how many bit, what is the bit depth of the image. So for example, a 1-bit image will just have 0 or 1. So you can just see two different colors here. But a 2-bit image would have 4 values, 2 power 2. So you see like 4 different shades in the image. And as the bit depth increases, the image clarity is higher. So generally, when you take images in a microscope, you can also set the depth at which the imaging is done. It's like some microscopes have 16-bit, some microscopes have 32-bit. Generally, it's better to go with a higher resolution imaging, and then later in the analysis, if you want, you can change the bit depth. So far, whatever we have been seeing here are single channel images, like they just have black and white, but you can also have different channel images like what we get from epifluorescence microscope imaging, or typically done after immunostaining or immunohistochemistry and so on. So here, instead of so in these images, there was just one number assigned, but in these RGB images, then each pixel will have 3 different numbers assigned, one for the red channel, one for the green channel, and one for the blue channel. Oh my, sorry, I'll share my slides again. Yeah. Then we looked at what are the important settings for dealing with multi-channel images. So you can, using split channel, you will get the 3 different images separately, red, green, and blue. And if you use merge channel, then, and if you have separate images, you can put them all together in one. And then again, you can change the LUT. Suppose this is a red image, but you want it to be displayed with the cyan setting. Then you can change the display with the LUT menu here. And there are also some lookup. This is a single color lookup table, meaning that it's all cyan. It's just bright and low cyan. But there are also some LUTs which have different values. So red are the high intensity pixels and blue are the low intensity pixels. But if you represent the image like this, then you also need to have a mapping of which color corresponds to which intensity or which pixel value. So that you have to go to analyze and tools, and if you select calibration bar here, then this will also display the colors that and the corresponding pixel values. Yeah, LUT bar, and then again brightness contrast was the same histogram that I showed. And then another type of imaging that data that we get from microscopy are these 3D images where the sample is imaged in different Z stacks, samples that have a height. And there we saw how to do images to stack. So for example, and for this was the, so all of these I was doing with the examples or sample images that are already in FIJI from here. So for this, I used the confocal series example. Yeah, so these are images which have both Z stacks and channels. So Z0 is the bottommost Z stack. Or let's say we just want to make, you're just interested in the red channel, which is C1. Now if you do image stacks, images to stack, then it becomes a stack. But yeah, right now these TIFF files already have the metadata about that it's a 400 pixel image completely, and 400 pixels correspond to 21.78 micron. You can also check this in the settings with Control-Shift-P shortcut. So these parameters are already set. I'm just sharing my screen again. Yeah. Properties, and then so if you want to show all of the 3D images as a single image, then you can do a Z project. And there are different methods of doing a Z project. So generally, whatever you want to do with these 3D images can be found in image stack. All the settings are here. And whatever settings you want to change with colors, all the settings are here. It will take a while, like maybe the first few times you might have to search where is the setting, where is that setting. But as you keep using it more and more, then you will know which setting is in which place. But for these 3D images, most of the settings are in image and stacks, and you can do a Z project. There are different ways. So one way is the average intensity. This can be used if all the Z stacks have useful information and you want to like show information from all the Z stacks, then average intensity would be good. Max intensity can be used if, say, you have an image which is more or less 2D, and it's just that it is out of focus in some plane. So not all the stacks have information, only the stack where the image is in focus has information, and that stack, if it is in focus, then it will also have the maximum intensity. So in that case, you can use a max intensity projection. And you can also, so this is typically an XY representation, but you can also view it in the Z. For this, I think this just has 3 Z stacks. I'll open the complete image. Focal. Ortho or orthogonal view. This whichever point you select, at that point it will show you the intensity across Z. So the bottom of this is the bottom of Z, and then as you go up, you can see the intensity in Z. So this kind of visualization will help you to know whether say you're looking at a protein, whether it is at the bottom basal side of the cell, or whether it is at the top or apical side of the cell. And there are also, again, other 3D volume views which you can do, for which more than FIJI, there are other softwares that are better with these 3D visualization. So one of it is Imaris Viewer. This I'm just showing you for these kind of 3D images. So here you can also get a nice volume view where you can really like move the sample in X, Y, and Z. There are also plugins in FIJI which can do that, but it's very slow and the rendering is also not as good as in this software. So this is Imaris Viewer. It is a free version. There is a better version called Imaris, in which you can also do a lot of analysis. This Imaris Viewer, you can just use it to view it in 3D. And again, here also you can get similar Z sections where you see the sample and at different height. This is the bottom of the sample, and as you go up, this is the top of the sample. So you can also, and these are, when I go from top to bottom, you can also see the imaging itself, how it changes. These are the original 2D images that we would acquire in the microscope. So this was about viewing 3D images. And then again, you have time-lapse images, where to open a series of images, you just have to select all of them and drag them into FIJI. So I'm just opening the first 10 images for now. And then when you do stack and images to stack, it will all combine together. So here you can see that there is one particle which is moving and then dividing. And before you do any kind of analysis, just change the properties. So this I did by doing Control-Shift-P. That is the keyboard shortcut. And these are not Z slices, these are time series. So I'm making it time. And I'm just roughly giving this value. Yeah, and then in these time lapse or any kind of stack, time stack or Z stack, typically the brightness settings that you set will be propagated to all the images. And then we also looked at how to use the ROI manager. Yeah. So here, suppose I want to measure the area of the nucleus of each cell. Then you can go to the freehand tool and just, right now I'm drawing it very roughly, but you can zoom in and draw it better. And now after you have finished drawing, sorry, the screen has stopped. Yeah. So once you're done with drawing this, you press T on the keyboard. That is the shortcut to add this to the ROI manager. Now you can similarly draw for all the nuclei, add it to the ROI manager. So here this is a kind of segmentation, but we are doing it manually. We are getting the outlines of the nucleus in this case, or we can also do it for the cell. But later in this course, we will also look at softwares that can do it on their own. We don't have to do it manually, because right now these are 4 cells. So we can still draw the outlines, but if there are many, then it will take a lot of time. So now these are the ROIs. And if I click measure, then the software will tell you what is the area of the ROI, what are the X and Y centroids, and so on. So for that, you can use this ROI and measure function. Also, to get this measurement, I pressed M. That is the keyboard shortcut. What else did we discuss? Then we also looked at what these different tools in the toolbar can do. Then we also saw how to open raw microscopy files from the different microscope softwares. I showed you CZI and from Zeiss microscope and LIF from Leica. OIR, I will check if I have any data in my hard drive. I can show it in the next class. But this is also similar. You just have to drag it into the FIJI window. So overall, this is the schedule that we will be planning for the course. So we did the first lecture today, which was just we looked at some basics of what is an image and some basic things that we can do with FIJI. And in the next lecture, we will go into how to show images in presentations and images or videos. Like you have to put the scale bar, and when you're putting two different images, they have to have the same brightness and contrast setting. So, and then you have to put a timestamp. And how do you annotate the images? Suppose you're making supplementary video for the paper, you will also have to write some, you might have to write some text. How do you add that? Yeah, it went. Yeah, anyways, right now I'm just on the slides. So, but anyways, I've shared my screen again. So, and then the third week will be a tutorial session where you can install FIJI on your own and do some of the things that I showed you today. We can again use these same sample pictures that I used as examples. So the third week will be a complete tutorial session where I won't be doing much. You will have to do things on your own. And if you have any doubts, then we can discuss. I'll be on the call. And then the fourth lecture, we will look at how to segment cells. Segmenting meaning drawing the outlines of cells, like to put it in a very simple way. So like, for example, in this class, we were doing it manually, but there is a software called CellPose, which can automatically detect cells. So one day will be for 2D segmentation, and the second day will be, the next week class will be for 3D segmentation. But if we finish 2D segmentation on the fourth week, then I don't think 3D segmentation will need a full class. So if there is not, sorry, it's not cellulose, it's CellPose. So on the fifth week, if there is more time after finishing 3D segmentation, then we can also look at other methods of segmentation. So this CellPose is an ML based method, but there are also other thresholding based methods of segmentation, which are useful when there is the nucleus channel that is quite easy to segment. And then again, the sixth week will be a tutorial, and this tutorial will be on the 2D and 3D segmentation. Here I will share some sample images that you yourself can try and segment it. So, and after you segment it, you can also get other measures of the cell, because once you have the cell outline, you will also get its measures like area, perimeter, what are the intensity values inside the cell, and so on. So we'll also discuss how to do that. And then the seventh week will be for cell tracking. So this is for time-lapse microscopy imaging, like this one that I showed, where we can see that the particle is moving and then it is splitting. So when you do tracking, you will be able to track it at different time points and say what is the speed at which it has moved, what is the direction at which it has moved. Right now, this is just one single particle, but you can also do it when there are many cells that are moving around. So that is about cell tracking. And then finally, the last will be a tutorial session for cell tracking. And if you have any other doubts, we can discuss that. And I was also mentioning in the beginning of the class that this is a course for image analysis for biologists. So it will, like, things will be simple here. You will just learn how to use the different tools that are already there. We won't be going into so much detail about how those specific algorithms work. Like, if, for example, if we learn cell pose, we won't really go into the nitty gritties of how exactly does cell pose work and so on. So if you're interested in these aspects, more of the algorithm aspects, then there are many other advanced image processing courses. But this is a basic image processing course where we just learn how to use the tools that are already there. Yeah, that's it for today. If there are any doubts, we can discuss. If not, we can meet in the next class. And this link I have, I have a folder where I have all the slides for the presentations. I have put it in the Teams chat. If you can't access it, let me know. I'll change the access settings. I think I had shared it with access to everyone. So yeah, it says anyone with the link can view it. So everybody should be able to access it. I can put it on the chat again for those who might have joined late. Denoising and upscaling. Maybe if there is time after any of the sessions, we can include that as well. Yeah. Okay. So how many of you have FIJI already installed? You can just raise your hand. Okay, some of you have. The others, if you want, even before the tutorial, you can install FIJI so that even in the lecture classes, you can try and along with me, you can do whatever I do. Otherwise, it's fine. You can also just do it in the first tutorial session. Okay. Anything else? Any doubts or anything? If not, we can just end this session and meet next week. Okay, I guess then that's it. See you all next week. Thanks for joining.
Yeah.